Hello everyone, welcome to BMI5730. I am Yan Zhang, the instructor of Module 4. In Module 4, we will focus on proteomics, both the basic technologies and biomedical applications. Today is the first lecture on the topic, Proteomics Fundamentals and Applications. Before all data generation and analysis, I always remind myself, your analysis is no better than the data you collect. People also say, garbage in, garbage out. That means it is very important to generate data with appropriate study design and quality control. Otherwise, the downstream analysis would be meaningless or misleading. Now, what is proteomics? First, we define proteome. The proteome is the entire set of proteins that is or can be expressed by a genome, cell, tissue, or organism. It is the set of expressed proteins in a given type of cell or organism at a given time under defined conditions. It can monitor the static state or equilibrium state of protein expression of a cell or tissue. It can also monitor the dynamic state. For example, when you have measured the protein expression at multiple time points. Proteomics is a large-scale study of proteins. Proteomics is the study of the proteomes and their functions. Why do we study proteins? Well, we have been looking at RNA levels, for example, from RNA-seq or microarrays. However, RNA levels do not always accurately predict protein levels. Proteins are the machinery that makes cells function, and many proteins are targets of common drugs. Sometimes only knowing the information contained in the nucleic acid sequence or DNA copy number cannot directly tell you the information needed in the clinic. In these cases, we do need to measure the protein levels. For example, in liver function tests, clinicians usually perform groups of blood tests that provide information about the state of a patient's liver. The AST enzyme and the ALT enzyme, their proteins, are used for biomarkers of liver injury in a patient with some degree of intact liver function. Serum ALT level, serum AST level, and their ratio are commonly measured clinically as biomarkers for liver health. The tests are part of blood panels. People have adopted proteomics technologies for biomedical and clinical studies. This figure shows the increasing number of proteomics publications over the years. These technologies are playing a more and more important role in our research, as well as in clinical studies. In proteomics, what do we measure? In the lab, for cell line-based experiments, we can measure different protein expression between different treatments or conditions. We can also measure different modifications, such as protein phosphorylation, in response to the stimulus. For different study designs, we can use targeted proteomics or shotgun proteomics. Targeted proteomics has gained significant popularity in mass spectrometry-based protein quantification as a method to detect proteins of interest with high sensitivity, quantitative accuracy, and reproducibility. While shotgun proteomics allows global protein identification as well as the ability to systematically profile dynamic proteomes. It also avoids the modest separation efficiency and poor mass spectral sensitivity associated with intact protein analysis. For protein profiling, we do protein or peptide identification and quantification. In animal experiments, it is more difficult to get conditions perfectly, more impacting factors, and sometimes the amount of protein sample we can get are limited. In the example of drug development, one big issue is that the right target may not be just genetically driven. Say the drug target is a protein, maybe an enzyme. We need to find the right drug for the right target in the right setting for the right patient. Here, EGFR, epidermal growth factor receptor, is the protein target of the drug. The drugs are EGFR inhibitors in lung cancer. This figure shows the comparison of using two different drugs in two different groups. On the left, in the EGFR mutation positive group, gefinitab works better than carboplatin plus paclitaxel in terms of patient progression-free survival. Since the Kaplan-Meier curve for gefitinib is above the curve for the other treatment. Well, when you look at the right, in the EGFR mutation-active group, we observe the totally opposite result. Gefinitab works worse than the other treatment in terms of patient progression-free survival, since the Kaplan-Meier curve for gefinitab is way below. Now you see, we need to have the right target. EGFR mutation positive or negative makes a big difference in treatment outcomes. What about the right setting? Here we use the examples in BRAF targeted therapy in cancers. BRAF is a gene that encodes a protein called BRAF. BRAF is a protein involved in sending signals inside cells and is involved in directing cell growth. It has been reported that the BRAF gene is mutated in some human cancers, including melanoma. There are drugs treating cancers driven by BRAF mutations. Vemurafenib and dabrafenib are two drugs approved by the FDA. Giovadis et al. Reported a case where a patient with double mutation metastatic malignant melanoma shows an overwhelming response to dabrafenib. By overwhelming, they mean the dabrafenib treatment has significantly surprisingly good response. Quote, almost surprisingly for the rapidity of the effect, 1 week later, the lesion on the shoulder has reduced its size by 60%, and 1 month later it has completely disappeared from sight. CT scan documented the astonishing clinical response. Another example is in treating BRAF-mutated colorectal cancer. Kopitz et al. Reported phase 2 pilot study of vemurafenib in patients with metastatic BRAF-mutated colorectal cancer. BRAF V600E mutation is seen in 5% to 8% of patients with metastatic colorectal cancer and is associated with poor prognosis. As we mentioned before, the drug vemurafenib is an oral BRAF V600 inhibitor, has pronounced activity in patients with metastatic melanoma. In marked contrast to the results seen in patients with BRAF V600E mutant melanoma, single-agent vemurafenib did not show meaningful clinical activity in patients with BRAF V600E mutant colorectal cancer. Comparing these two examples, we see that for different mutation status and different disease settings, the conclusion about the effectiveness and performance of drugs may be different. What's the right patient? This is related to something called precision medicine or personalized medicine. Like what's been said in OSU Cancer Center's advertisement, there's no routine cancer. We want to design targeted therapy for the patients. We want to find the most suitable drugs with the right doses that can best treat the patient. We want to maximize the effect while minimizing the side effect. Well, there's no such perfect drug without side effects. Now, in the past several slides, we have talked about the example of drug development. The drug target may be a protein, an enzyme. We can use proteomics technologies to monitor the changes in abundance or phosphorylation of the enzyme, comparing the drug-treated group with the control group. This is just one application scenario of proteomics. We summarize some applications here. We can do protein or peptide identification. We identify the proteins in the samples as many as possible. Then we compare the presence of them in different samples. Accompanying this, we can do protein or peptide quantification, or you might call it protein expression profiling. Besides the identities of proteins, we also want to know their abundance. We can further perform differential expression analysis to find significantly differentially expressed proteins. For the set of differentially expressed proteins, we can perform pathway enrichment analysis and associate them with functions. You can call this functional proteomics. Besides protein abundance, we may be interested in protein post-translational modification. We want to identify how and where the proteins are modified. For example, phosphorylated. This phosphorylation sites and phosphogroup abundance can be detected using specific mass spectrometry-based proteomics technologies. When we have expression or phosphorylation profiles of the proteins, we can generate protein-protein association networks. This network may be a co-expression network or a regulatory network, or others. We can use different data and methods to model them. Some even use proteomics to study protein structure. In structural proteomics, we may want to know whether the amino acid sequence of a protein confers its three-dimensional structure. From this, we want to determine how structure relates to function. Thanks for watching. See you next time.